Hello friends and welcome. Today we shall talk about tobacco mosaic virus. The main objectives of today's deliberation are: Number 1. To know the history, symptoms and disease cycle of tobacco mosaic virus (TMV). Number 2. To understand different sources of tobacco mosaic virus. Number 3. To know about the structure of tobacco mosaic virus. Number 4. To study mechanism of tobacco mosaic virus DNA replication. And finally, to know about the disease management strategies of tobacco mosaic virus. To begin with, let me give you a brief introduction about tobacco mosaic virus. Tobacco mosaic virus or TMV, it is a positive sense single-stranded RNA virus, genus Tobamovirus, that infects a wide range of plants, especially tobacco and other members of the family Solanaceae. The infection causes characteristic patterns such as mosaic-like mottling and discoloration on the leaves. Tobacco mosaic virus was the first virus ever to be discovered. Although it was known from the late 19th century that an infectious disease was damaging tobacco crops, it was not until 1930 that the infectious agent was determined to be a virus. It is the first pathogen identified as virus. Let us know about the history of tobacco mosaic virus. In 1886, Adolphe Meyer first described the tobacco mosaic disease that could be transferred between plants, similar to bacterial infections. In 1892, Dmitry Ivanovsky gave the first concrete evidence for the existence of a non-bacterial infectious agent, showing that infected sap remained infectious even after filtering through finest Chamberlain filter candles. Later, in 1903, Ivanovsky published a paper describing abnormal crystal intracellular inclusions in the host cells of the affected tobacco plants and argued the connection between these inclusions and the infectious agent. However, Ivanovsky remained rather convinced, despite repeated failures to produce evidence that the causal agent was an unculturable bacterium, too small to be retained on the employed Chamberlain filters and to be detected in the light microscope. In 1898, Martinus Beijerinck independently replicated Ivanovsky's filtration experiments and then showed that the infectious agent was able to reproduce and multiply in the host cells of the tobacco plant. Beijerinck coined the term virus to indicate that the causal agent of tobacco mosaic disease was of non-bacterial nature. Tobacco mosaic virus was the first virus to be crystallized. It was achieved by W. M. Stanley in 1935, who also showed that tobacco mosaic virus remains active even after crystallization. For his work, he was awarded 1/3 of the Nobel Prize in Chemistry in 1946, even though it was later shown some of his conclusions were incorrect. The first electron microscopical images of tobacco mosaic virus were made in 1939 by Gustave Cochet, Edgar Fancush and Helmut Ruska. In 1955, Hans Frankel, Conrad and Robley Williams showed that purified tobacco mosaic virus RNA and its capsid protein assemble by themselves to functional viruses, indicating that this is the most stable structure, the one with the lowest free energy. The crystallographer Rosalind Franklin worked for Stanley for about a month at Berkeley and later designed and built a model of tobacco mosaic virus for the 1958 World's Fair at Brussels. In 1958, she speculated that the virus was hollow, not solid, and hypothesized that the RNA of tobacco mosaic virus is single-stranded. This conjecture was proven to be correct after her death and is now known to be the positive strand. The investigations of tobacco mosaic disease and subsequently discovery of its viral nature were instrumental in the establishment of the general concepts of virology. Now, coming to symptoms of tobacco mosaic virus. The symptom is systemic mosaic type. The primary symptom on young leaves is faint circular chlorotic lesions appear with gradual vein clearing. This is followed by the development of characteristic systemic mosaic. With the maturity of leaves, abnormally dark green spots appear, which develop into irregular, crumpled blister-like areas, while the rest of the tissue becoming more or less chlorotic. Various degrees of leaf malformation like innations follow, and some leaves exhibit only a mild diffuse mottle. The development of symptoms is governed by many variable factors, of which the most important is the difference in virulence of the virus strains. For example, one strain of tobacco mosaic virus may cause yellow mottling on the leaves. A second may cause necrosis only, whilst a third induces a gross malformation. Another variable factor is the variety of plant affected. In flowers, petals show mosaic symptoms. Severe strains cause streaking of stem. The disease is seldom fatal to the host. Let us see the causal organism of tobacco mosaic virus. The typical tobacco mosaic virus is tobacco mosaic virus 1, Murmur tabaci-Holmes. The virus remains active in extracted host plant juice even up to 25 years. It's a very resistant virus, can stand desiccation for 25 years or more. It occurs in very high concentration in plant and its dilution endpoint is 10 to the power of -6. The thermal inactivation point of the virus is 90 degrees Celsius. The virus particles are rod-shaped, measuring 280 microns in length by 15 microns in width. The X-ray studies reveal that the virus particle consists of a number of protein subunits set in helical array with 49 subunits to one turn of the helix and 2130 subunits in one rod. The ribonucleic acid thread intertwines more or less centrally between the protein subunits. The cells of tobacco plants infected with tobacco mosaic virus are characterized by the presence of certain cell inclusions. They are two types of intracellular inclusions and intranuclear inclusion. The intracellular inclusions are X bodies and straight material of crystalline plates. The X bodies are amorphous, protoplasmic, more or less vacuolate inclusions, whereas straight material of crystalline plates give protein reaction. These crystals resemble the purified virus protein crystals. The intranuclear fibrous and crystalline inclusions are produced by a yellow mottling strain of tobacco mosaic virus. Coming to the disease cycle of tobacco mosaic virus. The virus perinates in infected tobacco plant debris, tobacco refuse from warehouses, cigarettes, cigars, pipe and chewing tobacco, and in perinating hosts which form the source of primary inoculum. The virus is disseminated from plant to plant by mechanical transmission, by handling tobacco plants during transplanting, through other field operations, and contact by man and cultivation implements. The virus enters the host tissue, multiplies very rapidly, producing disease symptoms. Now, let us know the source of virus. Tobacco mosaic virus overwinters in a number of ways, and these must be understood for successful control through sanitation practices. Infection of tobacco from overwintering sources of the virus is known as primary infection, and it includes number 1, tobacco products. All forms of tobacco may carry tobacco mosaic virus, so it is advisable that these products should not be used by workers, especially around greenhouses and during transplanting. Spraying plants with milk prior to pulling and transplanting will reduce the number of plants that become infected. It should always be used during transplanting in situations where tobacco use by workers cannot be prevented. Transplants should be sprayed until runoff with 1 pound of dried milk in a gallon of water immediately prior to pulling. Number 2. Tobacco trash. Tobacco mosaic virus will survive for years in dry tobacco tissue. So anything that may be contaminated with pieces of leaf, stem or root tissue should be cleaned prior to use in the crop. This includes floor trays that may have infected roots in the walls of the tray. Soilborne virus. Tobacco mosaic virus overwinters in infected stalk and root. Experimental data in North Carolina indicates that infection occurs at transplanting when a plant is pushed against a piece of virus-infected tissue. The number of transplants that become infected in this way will depend on quantity of overwintering tissue surviving. The virus will overwinter in dead as well as living plant tissue, but dead tissue contains less active virus than living tissue. Number 4. Other crops. Tomato, pepper, and eggplant are hosts of the tobacco mosaic virus, and in addition to not being used in rotation, should not be handled prior to working in tobacco. Fruit from these crops also contain active virus and should not be handled while working in tobacco. Number 5. Weeds. A number of weeds are known to be hosts of tobacco mosaic virus, but horse nettle is the only one found thus far to be infected. This weed is common where fluke-cured tobacco is grown and frequently is found infected with tobacco mosaic virus. The virus can be transmitted from infected horse nettle to tobacco mechanically, and this weed is suspected to be the source of tobacco mosaic virus found in many tobacco fields. The only way horse nettle can be eliminated as a source of tobacco mosaic virus is to eradicate it. Now let us know the secondary spread. Most of the mosaic plants in heavily diseased fields were infected by virus that was spread from a few tobacco plants infected from overwintering sources of the virus. Secondary spread can be reduced by removing primarily infected plants from the field or by cultivating in a manner to prevent spread of the virus. In seedling production, clipping is a very effective means of spreading tobacco mosaic virus. Seedlings should be scouted closely for tobacco mosaic virus at each clipping. There are a number of factors that must be considered when deciding if rogging in the field is feasible. So the final decision must be based on each situation. As a general guideline, however, the two most important factors are the time that primary infection occurred and the number of infected plants. Although primary infection can occur any time during the growing season, the most critical time is during transplanting. Plants infected at this time from any of the overwintering sources will show mosaic symptoms in 2 to 4 weeks. Generally, if fewer than 100 plants per acre are showing symptoms at this time, it is feasible to remove them. Rogging should of course be done prior to cultivation. Plants used to replant rogged plants frequently become infected from virus in the roots left in the soil when plants are removed, so replanting is not recommended. If too many plants are infected to make rogging feasible, secondary spread can be reduced by carrying out cultivation operations so that equipment does not touch the plants. Where this is possible, cultivation should be done when the plants are dry and preferably partially wilted because less virus transmission occurs under these conditions. It is seldom feasible or of value to rog plants after lay-by because most secondary spread occurs during or before this operation. Losses can be reduced by keeping a good supply of water to the crop because mosaic burn, the most damaging form of the disease, usually occurs only when infected plants come under water stress. Irrigation of a field containing a significant amount of mosaic may be worthwhile under conditions of moisture stress that would not be of value to rog after lay-by if fewer than 10 to 20 plants are showing symptoms by the time the crop is knee-high. This will reduce losses on the current year's crop somewhat and perhaps more importantly, prevent extensive spread to the remainder of the crop and thus overwintering of virus in stalks and roots. Now let us know about the structure of tobacco mosaic virus. Tobacco mosaic virus TMV is a particularly suitable object for studies on the physical and chemical structure of viruses and on the mutagenic effects of various treatments. The virus may be obtained in gram amounts and readily purified. Its biological activity may be assayed quantitatively by measuring the number of necrotic lesions which it produces on the leaves of suitable tobacco plants. Mutants may be detected by noting the characteristics of individual lesions or the visible expression of symptoms in systemically infected plants. The internal structure of tobacco mosaic virus has been determined largely by X-ray analysis, aided and confirmed by electron microscopy. The particle is a nucleoprotein containing about 5% ribonucleic acid RNA. The protein component consists of 2,200 subunits, presumably chemically identical, arranged in a helical manner to produce the rod-like structure of the virus. Embedded within the protein helix is the RNA, consisting of 6,600 nucleotides and appearing to follow the pitch of the helix of the protein subunits. Each protein subunit of molecular weight 17,500 consists of one chain of 158 amino acids, the complete sequence of which has been independently determined in Tübingen and in Berkeley. There appear to be no simple regularities in the sequential arrangement of the amino acids. Protein subunits alone, that's without nucleic acid, are capable of a characteristic reaggregation under suitable conditions of solution to form rods which are indistinguishable from those of the intact virus. The RNA component of tobacco mosaic virus can be isolated from the virus as a single polymeric entity with a molecular weight of about 2 million. The kinetics of the action of ribonuclease indicate that it exists as a single polynucleotide strand, in contrast to the double strand character of DNA. In solution, the RNA polymer shows evidence of coiling and folding, and this tertiary structure may be reversibly affected by changes of temperature and of ionic strength of the solvent. It appears then that the RNA of tobacco mosaic virus is a molecule consisting of one linear sequence of 6,600 nucleotides, a number far too great to allow a sequential determination of nucleotide arrangement to be made at this time. Some information about sequence is being gleaned from the products of degradation obtained by specific enzymes. Such degradation fragments have been chromatographed two-dimensionally and show a highly complex pattern which defies any attempt at sequential analysis. However, the pattern of RNA from tobacco mosaic virus is characteristic of this material and is easily distinguishable from that of the RNA of the host cell and of other RNA viruses. The function of the nucleic acid is now generally accepted as being primarily a genetic one. It has been shown that when RNA is carefully isolated from the virus by any one of several methods, it will produce the same symptoms of infection as will the intact native virus. The level of infectivity, however, is much lower than that of the virus from which it is isolated. It has been shown by meticulous assessment of the amount of protein material associated with the purified RNA that contamination by protein cannot be the source of the infectivity of the RNA. Indeed, the infectivity of isolated RNA is not reduced when the level of protein contamination is less than an amount sufficient to provide a single protein subunit per RNA molecule. Another bit of evidence for the absence of appreciable contaminant protein is the fact that the purified RNA is not in any way inactivated by antiserum against the viral protein. Since the RNA from tobacco mosaic virus may be isolated in highly purified form and is ineffective, it serves as a particularly suitable object for studying the structural basis of the transfer of genetic information. It has been found that infection may be initiated by the RNA molecule only in its intact form. Smaller fragments which can be isolated to some extent by centrifugation are inactive. Similarly, treatment by ribonuclease to the extent of causing only a single cleavage of the polynucleotide causes inactivation. On the other hand, the effects of changing the tertiary structure of the molecule are not in themselves inactivating. It appears then that the biological activity and genetic specificity are determined solely by the complete sequence of most or all of the 6,600 nucleotides within the RNA. Now, coming to the mechanism of tobacco mosaic virus RNA replication. The replication of tobacco mosaic virus RNA, like that of other positive stranded RNA viruses, consists of synthesis of a complementary negative strand using the positive strand as a template, followed by synthesis of progeny positive strands using the negative strand as a template. The negative strand will also act as a template for the synthesis of subgenomic mRNAs for the movement protein and the coat protein. During the early stages of replication, it's likely that progeny positive strands as well as acting as translational templates for synthesis of virus encoded replication proteins also serves as templates for amplification of the pool of negative strands. In tobacco protoplasts, synthesis of tobacco mosaic virus, negative strands ceases 6 to 8 hours after inoculation, whereas synthesis of positive strands continues for 16 to 18 hours after inoculation. Later in the replication cycle, after synthesis of negative strands has ceased and much coat protein has been synthesized, most of the progeny positive strands become encapsulated to form virions. Virus-specific RNA isolated from tobacco mosaic virus infected plants contains, in addition to genomic and subgenomic length positive stranded RNA, genomic length double-stranded RNA, designated replicative form, that is rfRNA, and a heterogeneous family of RNAs which are partly double-stranded and partly single-stranded, designated replicative intermediates, that is RiRNA. Molecules resembling Rf and Ri have also been detected following tobacco mosaic virus RNA synthesis in vitro using polymerase preparations from infected plants or protoplasts. In RiRNA, the number of positive strands on average exceeds that of the negative strands, and the structure of an Ri molecule has been interpreted as a single negative strand, to which are attached several positive strands of different lengths. Rf and RiRNAs are considered to be intermediates of RNA replication, or derived from intermediates of replication. An RF molecule could arise by initial synthesis of a negative strand on a positive strand template. The negative strand of the RF could then act as the template for production of progeny positive strands via Ri intermediates. If only one initiation event per negative strand template took place, an Ri structure would have only one single stranded, that is positive strand tail, and an RF would be regenerated after the completion of synthesis of each positive strand. Multiple positive strands in Ri structures would arise from repeated initiations of positive strand synthesis on a negative strand template before synthesis of the preceding progeny positive strand had been completed. During synthesis of tobacco mosaic virus RF in vitro systems with labeled NTP substrates, most of the label is found in the positive strand of the RF. This is inconsistent with a conservative mechanism, but is consistent with either a semi-conservative strand displacement mechanism or formation of RF by annealing of free positive and negative strands. Double-stranded RNAs corresponding in size to subgenomic mRNAs have also been isolated from tobacco mosaic virus infected plants. There is no evidence that subgenomic RNAs are self-replicating. Tobacco mosaic virus subgenomic RNAs are likely to be synthesized by internal initiation on genome length negative strand RNA templates, which requires subgenomic promoters containing sequences not found in the subgenomic RNA itself, as shown for pro-mosaic virus. Subgenomic double-stranded RNAs are therefore probably dead end products formed as a result of synthesis of a negative strand on a subgenomic positive strand RNA template. Let us know about the movement of tobacco mosaic virus in the infected plant. Tobacco mosaic virus uses its movement protein to spread from cell to cell through plasmodesmata, which connect plant cells. Normally, the plasmodesmata are too small for passage of intact tobacco mosaic virus particles. The movement protein enlarges the plasmodesmatal openings so that tobacco mosaic virus RNA can move to the adjacent cells, release the movement protein and host proteins, and initiate a new round of infection. As the virus moves from cell to cell, it eventually reaches the plant's vascular system, that's veins, for rapid systemic spread through the phloem to the roots and tips of the growing plant. Coming to the disease management. Number 1. Greenhouse management. It includes horticulture practices. To reduce infection of plants with TMV, all tools should be washed with soap or a 10% solution of household bleach to inactivate the virus. Tobacco mosaic virus contaminated soil should be discarded. To avoid transmitting the virus from an infected plant to healthy plants, the watering hose or watering can should not be allowed to make contact with the plants. Care should be taken to dispose of dead leaves and old plants because dry tobacco mosaic virus infected leaves can be blown around the greenhouse as dust, which can subsequently infect healthy plants if they are wounded. Cross protection. Inoculation of a mild strain of the virus onto young plants can protect them from subsequent infection by more severe strains of tobacco mosaic virus. This is a well-documented control strategy called cross protection that is successfully applied in greenhouse operations. Transgenic plants also offer alternative strategies for virus control. Number 2. Pre-planting options, greenhouse and field. It includes cultivars. Several tobacco and tomato cultivars have been bred to be genetically resistant to tobacco mosaic virus. Biotechnology. Genetic engineering techniques have provided scientists with the ability to express the tobacco mosaic virus coat protein gene in transgenic tobacco and tomato plants. This control strategy can safeguard the plants from infection by closely related strains of the virus. Elimination of inoculum. Under experimental conditions, it has been shown that tobacco mosaic virus can be inactivated when workers dip their contaminated hands in milk prior to planting. This inexpensive technique greatly reduces the incidence of disease. Seedlings that are known to be susceptible should not be transplanted into soil that contains tobacco mosaic virus contaminated root or plant debris. Number 3. Management in the field. Scouting for diseases. During the growing season, infected plants should be dug up, bagged and removed from the field. Rotation practices that include resistant plants or non-host crops also should be employed to reduce the amount of inoculum in the field. Number 4. Management at harvest and in storage. Tobacco mosaic virus can easily overwinter on the seed coat, thus providing an inoculum source for the next planting cycle. Therefore, it is important to treat tobacco mosaic virus contaminated tobacco seed with a 10% solution of trisodium phosphate for 15 minutes. Alternatively, tomato seed contaminated with tobacco mosaic virus can be incubated at 70 degrees Celsius for 2 to 4 days prior to planting. Both treatments will inactivate the virus that's on the seed coat, but should have little negative effect on seed germination. With this, we come to the end of today's lecture. Hope you have understood it well. Thank you and goodbye.